This is me, Daria, and ever since I've been this small, little bigger, yeah, this small, I've been leaving fingerprints on the TV, furniture, countertops, everywhere. But it wasn't until I was still small but less small that I discovered that the skin cells my fingerprints are made of can be used to make beautiful, limitless DNA profiles. Today we're going to be talking about DNA fingerprinting, and we'll start with the what. DNA fingerprinting is defined as the analysis of DNA from samples of body tissues or fluids. But why do we need them? They're most commonly used for matching crime scene biological evidence to a particular suspect or identifying biological ties and relationships. But before we get into the how, let's take it back. Way back. To 1984, when a man named Sir Alec Jeffreys found that the myoglobin gene in seals had the exact same genetic composition as those in the human genome. And that's when he coined the term DNA fingerprint to represent each and every human, or seal's, special genetic makeup. Now let's get down to business as we explore the steps on how scientists obtain these fingerprints in the first place. Step 1. Extract the DNA. It usually comes in the form of blood, cheek swabs, skin, semen, or hair, which are then treated with chemicals to get the DNA all by itself. Step 2. Restriction enzymes literally attach onto specific DNA sequences, creating breaks in the strands. Step 3. Gel electrophoresis. This cool device right here has an electrical current tug at the negatively charged backbone of the DNA fragments, causing the smaller fragments to move faster and the larger ones slower. And that brings us to step 4. Transfer to a membrane. A weak acid is used to break the DNA fragments on the gel into individual nucleic acids, which are then blotted onto nitrocellulose paper. And step 5. A radioactive probe sticks to the desired DNA fragments that the scientists actually want to work with, and because it's radioactive, it leaves a visible record on the paper. And that brings us to step 6, the X-ray, where the blot paper is exposed to X-ray film for at least a day to leave a visible record of the DNA fingerprint. And we're done. So what's the latest on this technology? Well, in 1994, the DNA Identification Act authorized the FBI to create CODIS, a cohesive forensic database that revolutionized the judicial system. DNA fingerprinting is also used for cancer treatment. When there's a tumor after an organ transplant, DNA fingerprinting is used to evaluate whether the tumor originated because of the donor or recipient, so it can be targeted and treated in the most effective way possible. And finally, groundbreaking technology like 23andMe gets mailed your spit and uses your fingerprint to connect you to your relatives and gives you a boatload of information about your ancestry. So, next time you see your fingerprint on your phone screen or window, take a second to think about how innovation means that residue could one day connect you to a long-lost relative or solve a criminal investigation. Because with DNA fingerprinting, we're really getting to a point where anything is possible.